Hello students, today's video is on DNA structure and function. Basically, DNA is found in the cells of all living things and contain all of the genetic information that makes you who you are. And every individual organism has unique DNA like a fingerprint. Most DNA is located in the cell nucleus, where it is called nuclear DNA, but a small amount of DNA can be also found in the mitochondria, where it is called mitochondrial DNA or mRDNA. DNA stands for deoxyribose nucleic acid, and it contains phosphorus in the form of phosphate. Deoxynucleic acid, it is a molecule that encodes the genetic instruction, and most DNA molecules are double-stranded helix, and each molecule consists of two long biopolymers made of simpler units called nucleotides. The structure of DNA is illustrated by a right-handed double helix with about 10 nucleotide parts put helical. Each spiral strand comprised of a sugar phosphate backbone and attached bases. It connected to a complementary strand by hydrogen bonding, non-covalent between base pairs, adenine with the thymine and guanine with cytosine. DNA has three main components, that is deoxyribose, that is a pentose sugar base. There are four different ones, and the phosphate. The bases are divided into two groups, pyrimidines and purines. Pyrimidines are made of one six-member ring, that is thymine and cytosine, and purines made of a six-member ring fused to a five-member rings, that is adenine and guanine. The rings are not only made of carbon, which is specific formulas and structures are not required for IB. The purine and pyrimidine bases of each strand faces inward towards each other. The bases are stuck perpendicular to the long axis of the double helix, and the helix has two external grooves. The narrow groove is called a minor groove, while the wide groove is called a major groove. The pairs of bases are always between a purine and pyrimidine, specifically the pairs AT and CG, which are the base pairs found by a char graph. There is hydrogen bonding between the bases, two H bonds between A and T, and three H bonds between G and C. Two chains do not have the same base composition, but two chains are complementary to each other. Such chains are called as complementary chains. Like first chain, that is ATACGCAC, and the second chain, that is TATGCGTCG. If we talk about the function of DNA, DNA has so many functions, but few of them are given below. Number one is replication. In this, double helix unwinds and acts as a template and forms double helix with the aid of DNA polymerase. Second function is encoding information, where a codon specifies a particular amino acids that which produce a particular protein. The third function is mutation or recombination. DNA plays a role in evolution of species. Like DNA can repair itself through recombination, and mutation occurs due to illegal base pairing, or both mutations and recombination either beneficial or create genetic diseases. Fourth function is gene expression. In this, cells from different tissues and organs look and behave differently. DNA can respond to produce a particular proteins by express a particular protein through transcription and translation. Transcription making RNA and translation making protein.